So the goal of this session is to introduce spatial phylogenetics to the broader conservation community, especially because of the fact that spatial phylogenetics has a lot of benefits that can help in describing and understanding biodiversity patterns on the landscape, which can help in conservation efforts and prioritization. And I hope that you will learn something new about this field that will be helpful in your research for conservation science, for practice and policy in the near and distant future. This is an interactive session, and so we want to ensure the maximum audience participation. So please feel free to type in your questions in the chat or to raise the hand button, especially when we get to the discussion section. At any point during the section, you can type in your questions in the chat or Q&A session. And you're also welcome to turn on your camera if you feel comfortable and the internet bandwidth permits, or otherwise, because we're going to be recording this session. And I have the pleasure to introduce you to the session organizers and presenters, starting with myself. My name is Israel Borokini, and I'm a Smith Postdoctoral Fellow in the Department of Integrative Biology, the University and Jepson Herbaria at the University of California, Berkeley. And I'm fortunate to work with my mentor, who is Professor Brent Mishler, who is the director of the University and Jepson Herbaria in the University of California, Berkeley. And also we have Matthew Klink, who is a postdoctoral fellow in the Department of Biology at the University of Vermont, an assistant professor in University of Nevada, Reno. Dr. Julie Allen is also one of us here today. And we have one of our colleagues as well, who is finishing up his PhD in the Department of Biology at University of Florida, Tal Kinser. Together, we are all going to be presenting different sections of this symposium, ranging from introducing the spatial phylogenetics, talking about facets, how we can use these methods for conservation prioritization. And then, of course, we have case studies and applications and general discussion period, which I hope is going to be enriching, and you're going to learn something new. We're going to learn from each other, and we can see how this symposium can, in fact, benefit the America the Beautiful, which is a 30 by 30 initiative of the government. And so without further ado, I would like to introduce Professor Brent Mishler, who is going to give us a broad introduction into spatial phylogenetics. Brent, please take it away.
Thank you very much. Go ahead with the next slide. So I'm going to try to introduce this complicated topic in as simple as possible of a way, and I'm going to start from scratch. So I don't mean to be insulting to anybody, but I just want to make sure that we're all on the same page with what we're talking about here before we go on to the more detailed examples that are coming up. So the cornerstone of this is the realization that biodiversity isn't about just species, that species are at best some particular level on the tree of life, that maybe a crazy hedge trimmer, crazy systematist has decided where the cutoff is, but there's certainly interesting phylogenetic information below and above the level that's called species. And so ideally you'd want to be able to look at the whole tree and not just species to make assessments of biodiversity. Next. Likewise, endemism isn't about species either. Any of these lineages, smaller or bigger than species, could be and are useful for conservation, and we need to consider their geographic distribution, how widespread they are. So the bottom line of this is that to really understand diversity and endemism, we need to look at the whole tree and not just species. Next, please. Not to insult anybody, but just to be sure we're on the same page with what a phylogeny is and isn't, I just wanted to show a really simple cartoon diagram right here. All that a phylogeny really indicates is two things, one of which is which of the terminals shared a more recent common ancestor, exclusive to them, that's not shared with the other organisms that are in the picture. And then the branch links, which have to do with some factor, which we'll be getting into a lot, but some factor that talks about what changes happened since the common ancestor. So next slide. Illustrated here is a simple characteristic. This could be any functional trait or a molecular mutation, but it has two different states that are present in different organisms. This next bit here shows it. There's this little interval of time that was shared between C and D and not with the rest of them. And then the next one shows that this particular trait would be inferred to have changed its condition on that branch, thus leading to a sharing of information between C and D. And that plus many, many other characteristics could be built up into a branch length, which would be one possibility would be the number of changes that are reconstructed to have happened on a branch. And also might be time, if you've scaled this to time. And we'll talk in more detail on the way you represent branch lengths on the phylogeny in a later, in the next section. These are called facets, different ways of arranging the branch lengths. And the last point there at the bottom, go back up. The last point there is that there's no sense to be made out of the order of the taxa across the top of the tree. A is not primitive to the others. Any of these nodes can rotate. So what the diagram is really showing is not any kind of linear order. What it's showing is a branching relationship among the terminals. Okay. So to measure biodiversity and endemism, we basically, in this new approach, we measure it right on the tree. And we're looking at the branch links that connect the terminal taxa that are present in some place. So the traditional metrics, species richness and endemism, are replaced by looking at the connections among the terminals that are in some place. And just as a quick visual, you can see that 14 terminal taxa in the left-hand phylogeny, those 14 are more closely related to each other than the set of 14 taxa in the other example where they're scattered on the tree, and so they're wide apart. And if you add up the green branches on the right, it's a longer path than the one on the left. So the one on the right would have higher phylogenetic diversity than the one on the left. And so phylogenetic diversity is defined as the branch links connecting the terminals that are present in an area to the base of the tree. And phylogenetic endemism is something I'll show you a slide of in a second, is that metric except taken on a tree that has some modified branch lengths where they're weighted by their range. And I'll show you what I mean in a minute. Next slide, please. So spatial phylogenetics is this field that was only made possible in the last few years because of the proliferation of data on the distribution of organisms and on phylogenetic algorithms and on big enough computers to do phylogenies of a whole biota at once. And we're finding it has lots of advantages, as Israel already pointed out, for looking at adding an evolutionary dimension to diversity assessment. It essentially takes that tree of life and makes it into a GIS layer that you can use on a map with other sorts of GIS data. Next. So here's just a quick calculation for PD, just to be sure that we're on the same page. Again, a cartoon diagram that has 5 terminals showing the branching relationships. And the PD here would be a reconstructed change along the branch. So a shorter branch has had fewer changes along it than another branch. And there's some shorter branches here and longer branches. If we take a selection of 3 of the 5 terminals, we'll get a different PD. So the one in the upper left is the maximum amount of PD we can get by picking 3 terminals. In this particular case, we're getting 90% of the tree. So PD is 0.9 for that one. And that's because we picked the longest branches on the tree. Whereas the middle right one is if we pick the short branches on the tree, the minimum PD we can get in this particular case is 0.55, just a little over half of the tree is present in a place where those 3 are. And then PD in this case would range somewhere between 0.55 and 0.9. And then there would be some average. And our randomization test is based on looking to see what the average would be. And then we look for outliers, which we'll get into in a minute. Next, I wanted to show how PE works. So if we can go to the next slide, where now this is the same cartoon tree, but what we've added here is range size in red. So range size here means how many pixels a particular taxon occurs in. So 1 is the maximum amount of endemism. It's only found in one place, and 5 in this case is the most widespread. It's found in 5 places. So if we take that tree and we divide each branch by its range, then hit the next click, Israel. It gives you a tree that would look about like this, where the really rare branch 0.25 has kept its branch length, been divided by 1, but the rest of them have been shortened. Hit the next click, Israel. That sort of blows up that part of the tree. And you can see the branch lengths on this new tree are the original branch length divided by its range size. This is what we call a range-weighted tree. So this is a different facet, has different branch lengths, but if you do PD on this range-weighted tree, you're going to get this measure that's called phylogenetic endemism. One more click. So PE is PD on a range-weighted tree. Next slide. We use 2 derived metrics in some of the stuff we're going to show you. So I want to explain those. Both of those are based on comparing the original tree that you had with a comparison tree where all branch lengths are equal. And that's in the lower right-hand side there, where all branch lengths are 0.125. So they're all a fair share of the length of the tree. And both of these metrics, RPD and RPE, are looking for places where there are a lot of long branches or short branches. So what we're doing is we're comparing the PD we get on our regular tree with the PD you would get on that comparison tree. And so the calculations, this is the same 3 trees. The RPD of the upper left one, for example, would remember it was 0.9 was the PD we calculated for that. And its PD on this comparison tree would be 0.75. So its RPD would be 1.2. In other words, there's some long branches in that one, and so forth. We also use that comparison tree in the denominator of RPE, which is an even more useful measure for conservation. So that's measuring phylogenetic endemism, as I explained a minute ago. And now we're looking for the place where the rare long branches are and the rare short branches. Next slide. This method that we call CANAPE, which is categorical analysis of neo and paleo endemism, uses RPE and a randomization test to find out where there are concentrations of branches that are both rare on the landscape and long, and rare on the landscape and short. And those are neo and paleo endemics, or centers where there are neo and paleo endemic lineages. So we can spot those on the map. And what's done, the upper left is the same old original tree you've been looking at this whole time, which happened to have a rare short branch and a rare long branch on it. We are making that range weighted, which we've already showed how we do. That's upper right is that original tree range weighted, which we've already shown. And what we're doing for RPE is now we have the comparison tree in the lower left, and we're going to range weight that, which is now shown in the lower right. And then next slide. The key thing to note here is that the long branch in the original data is longer on the original tree than it is on the comparison tree. Whereas the rare short branch taxon, which was in the original data, is longer in the range weighted version of the comparison tree. So you're going to be able to pick out, if you have enough of these together to lend significance in the randomization, you're going to find locations where there are rare short and long branches. And so I'll just follow up with just a quick example of how we did this with California. And then Matt will be returning to some California data in a little minute. But this is a published paper from 2017, so you can check it out if you want. But these are basically the raw patterns. On the upper two panels are raw patterns of species, richness, and endemism. And then on the lower two panels are the phylogenetic diversity and the phylogenetic endemism mapped. And particularly TR, which is taxon richness, and PD are quite similar. So you need to have a randomization test. You need to find places where the expected correlation between taxon richness and PD is different than expected. So just to show you what these results look like, next slide. We have a, here's what the trees look like, just to give you an intuition for this data set. So above left A is what the original tree looks like with molecular branch lengths. And B is what it looks like range weighted. And then C is the comparison tree with all equal branch lengths. And then D is what that tree looks like range weighted. So when I show you RPD, it's going to be comparing upper left, upper right. When I show you RPE, it's going to be comparing the upper lower left to lower right. Next slide. When we randomize PD per se, we notice places in California, such as up in the northern Central Valley. You can see there and along the north coast, where there are where there's less PD than there should be with that selection of that number of taxa. And as you can see here, that may have an ecological explanation, may have other explanations as well. But a place that's in blue in the upper right map is a place where distant relatives are living together, whereas the areas in red in the upper map are where the PD is significantly low or close relatives are living together, which also may have an ecological explanation if there's certain traits that help you live in the desert. And if those are phylogenetically conserved, then there's going to be a lot of those in the desert, which you do see here. RPD, remember, is slightly different. That's showing where there's concentrations of long or short branches. And again, northern Central Valley and then the whole north coast is amazing in that way. And also some areas in the Channel Islands and down in Southern California. So there could be some ecological connotations, but it may also be biogeographic as well. You could have refugia, where there are taxa that have been hanging around for a long time. You're going to end up with a collection of long branches. So you can have sort of biogeographic effects that show there. And then one last slide, just to show you what the canopy looks like. On the left-hand side is where the rare long and rare short branches show up. So there are centers of neoendemism out in the Death Valley and out along that area. And there are centers of paleoendemism in the Sierra foothills. And then the purple is where you have mixed paleo and neoendemism, which in California, we have mechanisms that cause both. So we have regions such as the deserts that have a lot of both. And then just the one on the right is a topic we're not covering very much today, but just to point out that there are phylogenetic methods of looking at turnover. So what I've been talking about so far are alpha diversity measures, but there are beta diversity measures that would let you compare these different sites. And the branching diagram on the far right is a cluster diagram that's showing how those centers of endemism relate to each other in terms of what part of the tree they share. And we get some very interesting information out of that kind of turnover metrics as well. So if you're interested in this topic, it would be something you'd explore further would be beta diversity measures based on phylogeny. Next slide. So here we'll pause for any questions just about this kind of stuff, the metrics. Does it make sense? Why do it? If any of these questions are appealing to you, we can address these. These are something we thought of, or you're welcome to put in the chat any other questions that you have. Can everyone see why it does make a difference? One question I often get is, why do this? Like, what does it add to the regular species-based analysis that we're used to? So the answer to that one is you get the historical dimension. You can actually separate these interesting cases of long centers where there's long branches grouped together, or centers where there are short branches grouped together, which, by the way, may indicate places where diversification is happening. That's one implication of having a bunch of short branches in an area, is that they're young branches and potentially being generated in that place. Matt's going to talk more about that later. So we think it is. It is a little bit of extra work. You got to add a phylogeny. But these days, the phylogeny usually turns out to be kind of the low-hanging fruit. It's, you know, not the most difficult part of one of these analyses. It's usually the spatial data. And people often ask, well, what if you don't have a complete phylogeny? Like, I'm working on a group that isn't studied very well, and I don't have much of a phylogeny. You can do at least an initial analysis like this, even if your phylogeny only had a certain fraction of your actual diversity. The analysis I showed you of California plants only had about 1,000 and some terminals in it, whereas we have more than 5,000 species ranked taxa in California. So that was based on a sampling of terminal lineages. And we got patterns which I hope will be supported when we add more data, but that's kind of science. The randomization, the way we do it is pretty simple and unconstrained. We just take the distribution of the occurrences of a taxa off the map and put them back on randomly anywhere in the map, with the constraints being that the range size of a taxon stays the same, number of pixels, and the amount of diversity, number of pixels, stays the same in each grid cell. And then we just recalculate these metrics again and again and again after that kind of randomization spatially, and then we determine whether the observed measures are outliers in a two-tailed test. So that's how that randomization works.
Hey, Brent, can I ask a question?
Yep.
So are you aware of any policy measures that have taken into account any of these spatial phylogenetic metrics into thinking about areas of conservation, areas to conserve?
Yeah, we've had some success in California. Matt's going to talk about a couple of these cases in a few minutes. Cool. But it's hard to explain, but once people kind of get what's going on, it actually is quite appealing for conservation prioritization.
Yeah, it's neat. I mean, it just seems like a really different way. You know, obviously I think about this stuff, but it's a different way rather than just saying number of species is important, but understanding, you know, how those communities have come together is a process that's important to conserve as well.
I think that's a really important point that we're talking about. We're sort of moving from pattern to process a little bit when we can add the evolutionary dimension.
Yeah.
So that's almost exactly 20 minutes. So I think we should move on to Israel section. I think I'm supposed to introduce him, but you've already met Israel.